Welcome to our session on food poisoning, a common public health problem that every physician will encounter. Today we'll explore the critical distinction between intoxication and infection as causes of food poisoning, and learn to recognize different bacterial agents based on their clinical patterns. Understanding incubation periods, symptom patterns, and associated foods helps us identify the causative agent and implement appropriate outbreak control measures. Let's start with a classic case of food intoxication. 24 people became ill within 3 hours after eating a meal at an office party. Let me emphasize that timeline, within 3 hours. This extremely short incubation period is a crucial diagnostic clue. All had nausea, most had vomiting, and several had crampy abdominal pain. Notice that vomiting is the predominant symptom, not diarrhea. This pattern is characteristic of certain types of food poisoning. 3 individuals were sick enough to seek emergency care. Importantly, all 24 people had been in good health before the party. This rules out other causes. Now let's examine the food exposure. The day before the party, a food preparer purchased a 5-kilogram pre-cooked packaged ham. She baked it at home and transported it to her workplace, a large institutional kitchen. Here's the critical step. She sliced the hot ham in the kitchen. The ham was then served cold at the party the next day. Let's analyze what went wrong. The ham was handled when hot, perfect for bacterial growth if contamination occurred. It was then held at room temperature overnight, before being served cold. This time temperature abuse allowed bacteria to multiply and produce toxin. Based on the 3-hour incubation period, predominant vomiting, and the food vehicle sliced ham handled by food preparers, this is classic Staphylococcus aureus food intoxication. Let's understand the fundamental classification of food poisoning. There are 2 main types, intoxication and infection. Understanding this distinction is crucial. Intoxication occurs when you ingest a toxin that was produced by bacteria in the food before it was eaten. The key point: the bacteria made the toxin while growing in the food, and you're consuming preformed toxin. The classic example is the heat-stable enterotoxin of Staphylococcus aureus. Intoxication has several distinguishing features. First, it has a shorter incubation period than infections, often just 1 to 6 hours. Why? Because the toxin is already present and doesn't require bacterial multiplication in your gut. It acts immediately. Second, symptoms are typically abrupt and severe, with prominent vomiting. The toxin directly stimulates the vomiting center in the brain. Third, intoxication may involve extraintestinal symptoms. The classic example is botulism, caused by Clostridium botulinum, where the neurotoxin causes neurologic damage and paralysis, not primarily gastrointestinal symptoms. In our case scenario, the 3-hour incubation period and predominant vomiting clearly indicate intoxication, not infection. In infection, bacteria are ingested along with contaminated food. These viable bacteria must then multiply in your gastrointestinal tract to cause symptoms. The classic example is non-typhoidal salmonella. Alternatively, bacteria may be ingested and then release enterotoxins in the gut as they multiply, resulting in gastrointestinal symptoms. Clostridium perfringens works this way. The bacteria produce toxin in your intestine, not in the food beforehand. Key differences from intoxication. First, longer incubation periods, typically 6 to 72 hours, because bacteria need time to multiply before causing symptoms. Second, symptoms develop more gradually as bacterial numbers increase. Third, diarrhea is often the predominant symptom rather than vomiting, because the toxins are produced in the intestine. Let's go through each agent systematically, focusing on the pattern recognition that helps diagnosis. Starting with intoxications. Staphylococcus aureus. Incubation period 2 to 4 hours. Primary symptom is vomiting. Characteristic foods are meat, custard, and salad. Basically any food handled extensively by food preparers and held at room temperature. This is exactly what happened in our case. Bacillus cereus. Incubation period 1 to 2 hours. Causes both vomiting and diarrhea. Classic association with rice, meat, and vegetables. The vomiting form is particularly associated with fried rice that's been kept warm. There's also a diarrheal form with longer incubation. Clostridium botulinum. Incubation period 12 to 72 hours, much longer than other intoxications. The key symptom is neuromuscular paralysis, not typical gastroenteritis. Classic foods are improperly canned foods, particularly those with neutral to alkaline pH held at room temperature. Botulism is a medical emergency. Now the infections. Salmonella species. Incubation period 6 to 48 hours, typically 12 to 24 hours. Primary symptom is diarrhea, often with fever and cramps. Characteristic foods are poultry, eggs, and meat. Remember our previous lecture on Salmonella gastroenteritis. Clostridium perfringens. Incubation period 9 to 15 hours. Causes profuse watery diarrhea. Classic association with meat and poultry, particularly when prepared in large quantities and held warm. Often called cafeteria germ, because of outbreaks in institutional settings. The pattern is clear. Short incubation with vomiting equals intoxication. Longer incubation with diarrhea equals infection. Bacillus cereus is a spore-forming, gram-positive rod found ubiquitously in soil and food. The spores survive cooking, which is why properly cooked food can still cause illness. There are two clinical forms. The emetic or vomiting form has a short incubation period of 1 to 6 hours. It's caused by a preformed, heat-stable toxin called cereulide. The classic scenario is fried rice syndrome. Rice is cooked, held at room temperature, allowing B. cereus spores to germinate and produce toxin, then fried briefly, which doesn't destroy the heat-stable toxin. Symptoms include severe nausea and vomiting, similar to S. aureus food poisoning. The diarrheal form has a longer incubation of 8 to 16 hours. It's caused by enterotoxins produced in the intestine after bacteria multiply. Symptoms include watery diarrhea and abdominal cramps. Key foods associated with B. cereus include rice, pasta, meat, and vegetables. Prevention focuses on proper refrigeration and avoiding prolonged holding at room temperature. C. perfringens is a gram-positive anaerobic sporing bacilli that's widely distributed in soil, water, and the intestinal tract of humans and animals. Its spores are heat-resistant and survive normal cooking temperatures. The pathogenesis works as follows. Meat or poultry is cooked, which actually activates the spores. If the food is then held at warm temperatures rather than properly refrigerated, the spores germinate and multiply rapidly. When consumed, the bacteria sporulate in the small intestine, releasing an enterotoxin during sporulation. The enterotoxin causes fluid accumulation in the intestinal lumen, resulting in profuse watery diarrhea. Clinical features include incubation period of 8 to 16 hours, sudden onset of watery diarrhea and abdominal cramps, usually no vomiting or fever, and symptoms lasting 12 to 24 hours. C. perfringens is often called the cafeteria germ or church supper germ because outbreaks commonly occur when large quantities of meat are prepared in advance and held warm. Let's consolidate this information into practical clinical patterns that help you identify the causative agent. Timeline matters. If symptoms appear within 6 hours, think intoxication, S. aureus or B. cereus emetic form. If symptoms appear after 6 hours, think infection or B. cereus diarrheal form. Symptom pattern matters. Predominant vomiting with minimal diarrhea suggests S. aureus or B. cereus emetic form. Predominant diarrhea suggests salmonella, C. perfringens, or B. cereus diarrheal form. Neurological symptoms suggest C. botulinum. This is an emergency. Food vehicle matters. Custard, cream-filled pastries, sliced meats handled extensively. Think S. aureus. Fried rice held warm. Think B. cereus. Undercooked poultry or eggs. Think salmonella. Large quantities of meat held warm. Think C. perfringens. Home canned foods. Think C. botulinum. For outbreak investigation, always document the common food source, precise incubation periods from all affected individuals, symptom patterns, and food handling practices, including time-temperature abuse. Remember, food poisoning is preventable. The key control measures are proper cooking temperatures, rapid cooling and refrigeration, avoiding prolonged holding at room temperature, good personal hygiene for food handlers, and proper food storage. Understanding these patterns allows you to make rapid clinical diagnoses, implement appropriate treatment, usually supportive care with rehydration, and most importantly, identify and control outbreaks to prevent additional case.